Next video clip for Chapter 6, we're going to talk about the study of bacterial growth. And some of this is pretty straightforward information. There is some new terminology in here, though. All right, so growth of bacterial populations. So remember when we said when we were talking about microbial growth is referring to an increase in population. And bacterial populations can generally increase pretty quickly. Now, there's a lot of variation out there in the world of bacteria, but in general, bacterial cells can divide pretty quickly if they're in an optimal environment. So all these things that we've been learning about, like temperature and pH and oxygen requirements and so forth, if a bacterial organism is in its optimal environment, the bacterial cells can generally divide pretty quickly. Bacterial cells divide through a process called binary fission. So binary, 2, fission to split. So one cell splits into 2 daughter cells. 2 daughter cells split into 4, 4 into 8, 8 into 16, and so forth, and so on. And as far as the steps, what actually has to happen? All right, so we've got a very simplified bacterial cell here. This is supposed to be a young cell at step number 1. The little purple squiggly thing in there, the chromosome, the DNA. Remember, the DNA are the genetic blueprints, like the recipe books that tell us how to make cell parts and allow the cell to operate and maintain itself. So if a cell is going to divide, each of the daughter cells must have a copy of the chromosome or chromosomes. Some bacteria have more than one. So the first thing that has to happen is the DNA needs to divide one copy of the DNA or the chromosome into 2. And then you create a division between those 2 new copies. So that division that's forming is called a septum. Septum is another name for a wall or a separation. And then once that's completed, you've now gone from 1 cell to 2. So pretty straightforward. Now in previous biology courses or in anatomy and physiology, you may have studied the process of mitosis. So mitosis is a more complex eukaryotic process that involves duplication of all of the chromosomes in the cell, and then making sure that each daughter cell gets a copy of all of those chromosomes. Or 2 copies, right? Human cells need 2 copies of each of your chromosomes. So you got to make sure, like with a human cell, how many chromosomes do your human cells have? They're supposed to have 46, so you got 2 copies of chromosomes 1 through 22, and then you have your 2 sex chromosomes, XX or XY. And so when one of your cells divides into 2, all 46 of those chromosomes have to be duplicated, and then you got to make sure that each daughter cell gets 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3. You don't want to have a random mixture of those 46 different chromosomes, because you got to make sure that each daughter cell gets a full set of genetic recipes. So mitosis is a complex process that makes sure those chromosomes get divided up into the daughter cells evenly. With bacteria, they generally only have 1 chromosome, so the process is not as complicated. So bacteria don't do mitosis. They divide through this process called binary fission. All right, another feature here is the time. How long does it take for 1 cell to divide into 2, then 2 into 4? That's called your generation time. Often it's very short for bacteria, so the example here is 30 minutes. So that means 1 cell divides into 2 into 30 minutes, 2 into 4 by 60 minutes, by 90 minutes 4 have gone to 8, and by 120 minutes 8 have gone to 16. And so you can kind of see where we're going from there. Okay, so now a lot of microorganisms, especially bacteria, yeast, do this as well, can explode in population, and it's because they undergo something that's called logarithmic growth or exponential growth, and they have these short generation times or reproduction times. So let's think about this. So 2 cells to 4 cells, 4 cells go to 8, 8 to 16, 16 to 32. If this is happening every 20 minutes, 20 minutes, 20 more minutes, 20 more minutes, because some bacteria can divide every 20 minutes. That's the generation time. If you've got lots of nutrients, optimal conditions for that cell. let's say in a theoretical bacterial culture, you put in one cell, optimal conditions, cells are dividing every 20 minutes. At the end of 7 hours, you're going to have 2 million cells in that culture. So 1 goes to 2 million after just 7 hours. And if you could maintain that optimal environment, after 24 hours, you would have 10 to the 21st cells. So that's a 1 with 21 zeros after it. Now, that's on the theoretical side. In reality, when you have a liquid culture over here with bacteria in it or yeast in it, they're going to use up the nutrients. That's a closed culture, a closed system. So they're going to use up the nutrients. They're making wastes just like our cells do. Those wastes build up and become toxic. And so you're not really going to be able to maintain ideal conditions in that little glass tube for a 24-hour period of time. But the main idea here is that microorganisms can grow explosively, and that's called logarithmic or exponential growth. Those are both mathematical terms. We've talked before about how turbidity in a liquid culture indicates that the culture, the organism is actually growing in that culture. And when you see a broth culture like this in the laboratory that looks cloudy, you can feel confident that there are at least trillions of live bacterial cells in there. Same thing inside our bodies. Microorganisms, if they have ideal conditions, can grow explosively. So that's why you can get exposed to a fairly low number of bacteria or some other type of microorganism. And if they're experiencing nice ideal conditions in your body, they can increase their populations pretty quickly. And once that population builds up to a certain point, they're going to start damaging you and cause problems for you. And over here, this is actually an image. This might look familiar to you from your A&P classes. In purple over here, this is actually the lining of the intestinal tract, the small intestine. So this is the wall. This would be your inside lining right in here, and down in here you're actually in the wall of the intestine. And out in here, this represents the space inside the intestine and also right along the lining. So all this darker purple stuff that you see right in here, bacteria and food inside there. So lots and lots and lots of bacteria in the intestinal tract. All right, so when you grow a microbial culture in the laboratory, and we're mainly thinking about bacteria and yeast, little single-cell fungi in this case, the growth pattern that you would see tends to follow a characteristic, what we call a growth curve. So again, we're thinking about a culture that's like a closed liquid tube like this. So you're not replenishing nutrients, you're not pumping out the waste or anything like that. It's just a closed culture with a limited number of nutrients. And the microbes are in there bathing in their own wastes for the most part. So when you set up a new culture, a closed culture like this, what we're looking at here on this graph, x-axis, this is time, and the time doesn't necessarily have to be hours. Or don't take the hours too literally, because this is going to vary for different kinds of bacteria and yeast that you might grow. And then on our y-axis over here, these are population numbers. This is a logarithmic scale, so where you see a 2, that actually means 10 to the second power, which is 100. The 4 here means 10 to the fourth power, which is 10,000. The 6 here is a million. That's 100 million, 10 billion. So it's not a typical regular scale like you might be used to. So at time 0 right here, this is where you are doing a transfer like you guys did on the lab simulations. You're transferring bacteria from one culture or maybe from a patient sample into a new one. And then you start sampling your new culture to measure the population of bacterial cells in there. And you would find that for a few hours after you set up a new culture, there wouldn't be much of a change in the population. And that's called the lag phase. So during the lag phase, the bacteria are adjusting to their new culture medium. because not all culture media are the same. So maybe you put them in a new culture media and the nutrient mixture in there is different. So we need a lag phase for those organisms to adapt to that new culture medium and those new nutrients that are in there. Once they adapt, though, you're going to have optimal growth for a while. This is your exponential or logarithmic growth phase. So the population is going to increase very, very rapidly. The culture is going to become turbid. Then you're going to hit a stationary phase. This is where your population evens out and kind of remains stable. And you do have cell reproduction going on in there, but you also have cells that are dying as well. Your nutrients are starting to run out. Your wastes are starting to build up. So you no longer have optimal conditions in there. So that's called the stationary phase, which is maintained for a while. And then finally you have a death phase. So eventually you're going to get to the point where the nutrients are being used up, the wastes are building up, and the cells begin to die. And so if you were measuring the population, it would actually start to drop off, and that's called the death phase. Now, for a while, if you just kept incubating that culture, eventually almost every cell in that culture would probably die. So what you'll see in a real laboratory, you grow a culture, but let's say you want to maintain that culture for a longer period of time because you might want to use it as a source to set up new ones. So instead of leaving them in the incubator, what you do is you shift them over to the refrigerator. So some students are surprised when they work in a real microbiology laboratory at first because you grow these cultures up and then you stick them in the fridge. Well, the refrigerator doesn't kill them. It just slows down their metabolism and prevents them from overgrowing in that culture and going into the death phase. So you will see in real microbiology laboratories that stock cultures are maintained in a refrigerator. And that prevents overgrowth, prevents the culture from reaching the death phase. That doesn't last indefinitely, though. That doesn't mean you can store cultures in a refrigerator forever and expect them to remain viable. There are limits to that. Generally in the lab, we have certain organisms for our face-to-face microclasses that we grow and use, and we try to passage those cultures, set up new cultures about once a month to make sure they stay fresh and viable. All right, so in your textbook, I just mentioned measuring populations of bacteria, and I am asking you guys to read that part of the textbook, and you will have a few homework questions on that, especially in the Chapter 6 Learn Smart Book reading activity, just so you've been exposed to some of that information and are familiar with it. But I'm not going to expect you to know details on how these microorganisms are, their populations are measured in the laboratory for testing purposes. All right, so again, why is understanding bacterial growth important? Lots of reasons from a clinical standpoint. Most of us are interested in clinical topics in this class. One of the reasons why bacteria are so good at causing disease is because they can grow very quickly, so they can overwhelm your immune system before your immune system has a chance to fight them off. And as we'll see when we get later into the semester and we start talking about transmission of microorganisms from one person to another, if you're infected with bacteria, they kind of follow a growth phase like that as well inside the body. And your symptoms that you have follow a similar type of pattern. So we're going to see that when we get into Chapter 11. And when would you be more likely to shed these microbes and spread them to other people? Hopefully you're thinking when the population is at its highest, you're going to have more of those microorganisms there to shed. And as I've mentioned already, a lot of times in a micro lab, the job there is to identify, diagnose an infection, what organism is causing the problem with the patient. And so current techniques, you have to grow the microbes in the laboratory, so we have to understand what their physical requirements are, what nutrients they need. We need to know which ones are fastidious, so we make sure we supply them with all of their needed growth factors to be able to survive. Also, when we get into Chapter 10, we're going to study antimicrobial drugs. And as it turns out, quite a few drugs only work on actively growing microorganisms. Handling is important as well. Later in the semester, we're going to get into into more detail in Chapter 11 on what parts of the body naturally have microorganisms on or inside them. And it's really important that you know where microbes naturally grow on or inside the body, because you've got to think about that when you are collecting samples from patients for various reasons. For testing purposes or for culturing purposes, you want to make sure you're not contaminating those samples with unwanted microorganisms from the patient's body or from your body. So you need to keep those things in mind as well. So proper clinical handling is always a big time important topic for this class, and you guys are going to have that information preached to you as you go through your health sciences classes. All right, so that actually is going to conclude Chapter 6. So we've talked a lot about the different requirements for microorganisms to grow, and I think a real key topic from Chapter 6 is this diversity, this very amazing, diverse world of microbes and their different lifestyles and adaptations. In Chapter 7, we're going to get into more details on microbial metabolism. So when we say microbes are harnessing energy from their chemical nutrients, how are they doing that? How does that all work? So we're going to delve into a little bit more detail there, and you may have covered some of this in previous classes. Some of you, this might be your first somewhat in-depth exposure to it, but that'll be the topic for Chapter 7.